Welcome everyone to the Center on Parenting and Opioids monthly webinar series. We're really delighted today to welcome Dr. Adrianne Huxtable to present with us today. Adrianne is an associate professor in the Department of Human Physiology at the University of Oregon, and she comes to U of O by way of Canada, where she did her undergrad and graduate training, and then came to University of Wisconsin-Madison for a postdoctoral fellowship before starting at the U of O in 2015. It's a little bit unusual for us to have a presentation focused on animal research. And so I just want to say a few words about why this is really important and helps foster a translational understanding of the work of the Center on Parenting and Opioids. So what Adrianne will speak to us a little bit about today is things having to do with early life stressors and maternal opioid use and how those factors can contribute to kind of lasting effects at the physiological or systemic level, all the way into adulthood in animal models. And what we can learn from animal models is a little bit different from our research with
Ashley, I lost the audio. I don't know if everyone else did.
Yeah, I did too.
Just as she was getting to all the interesting things and benefits of animal models, it cut out for me.
Of exposures, for example, to opioids or any of the contextual or environmental factors. So while this may seem a little bit distant from our typical presentation on moms and studying substance use in moms and families, there really is direct relevance for the work of our center, and in particular thinking about how we can bridge work from the basic sciences from animal models into the human world and interventions and prevention science related to the content of our center. So with that, I will turn it over to Adrianne. I apologize that I'm not able to be here today due to a conflict with some student activities, but I am delighted to have you here, Adrianne, and thank you for joining us.
Great, and we'll turn it over to you, Adrianne, when you're ready, if you have slides. And yeah, thanks so much for being here.
Yeah, thank you all so much for the invitation. It's really exciting to be able to be here. Hopefully you can now see my slides. I'm hoping anyway. Okay, great. And I was talking to Ashley just in advance. I'm really awful at monitoring a chat while I'm talking. So if there are questions, please feel free to put them in the chat, and Ashley's going to help me try to monitor it as I go. And thank you to Leslie for that really kind introduction. And I definitely understand that some of the work I'm going to be presenting today is different than what normally comes up in this group, but hopefully I can make you a little bit more interested in the animal models and in the respiratory system as a whole. So I'm going to focus on some of our current work that we are conducting here at the University of Oregon. And really, our research group is focused on studying respiratory neurophysiology and plasticity. And if none of those words mean anything to you, or you have no idea why you should care about them, that's totally okay. We'll spend a little bit of time talking about the respiratory system and respiratory control networks. So hopefully you can start to think maybe about them in the future. Over the last few years, as Leslie mentioned, we've become particularly interested in the effects of early life stressors and how that can influence development of neural pathways that control breathing. We're interested both in the acute effects of these early life stressors, but also the potential for there to be lasting consequences on neural activity and behaviors such as breathing. I'm going to focus today on two early life stressors. The first are the effects of neonatal inflammation. And then I'm going to move into a topic that I'm sure is of interest to many of you, looking at the effects of maternal opioids. What we hypothesize is that these early life stressors lead to changes and impairments in developing neural networks that control breathing. And we think that they do this by altering how distinct cell types within the central nervous system communicate with each other to ultimately change neural activity and change behaviors such as breathing. So why are we interested in breathing? Well, breathing is vital for keeping you alive. It is essential to maintaining homeostasis in the body by maintaining levels of oxygen, carbon dioxide, and pH. Breathing is also a motor behavior, but unlike many other motor behaviors, it has to be rhythmically active continuously throughout life to maintain homeostasis in the body. So the respiratory system has to be extremely robust, but it also has to be highly adaptable. It has to respond to the ever-changing internal and external environmental conditions, including those associated with development and aging, but also in response to disease and injury. There are many different components to the respiratory system. If you are to Google the respiratory system, which I know we're all doing in our spare time, you're probably going to find a lot of diagrams that look like this. Diagrams that highlight the diaphragm, the main inspiratory muscle that permits inhalation, the lungs, and the upper airways. But I would argue that most of these diagrams leave out the most interesting component of the respiratory system, and that's the central nervous system. So our lab is really looking at how the brain and spinal cord control all of these respiratory muscles that allow you to breathe. In specific, there are essential respiratory networks within the brainstem and the spinal cord that are really responsible for innervating all of these respiratory related muscles to allow you to breathe. If we look at a slightly more simplified version of this diagram, we're particularly interested in one area of the brainstem known as the medulla, as well as the cervical spinal cord. The cervical spinal cord contains motor neurons, and their axons are what form the phrenic nerves, which innervates the diaphragm, that main inspiratory muscle. If we look even more in detail into these central respiratory control networks, we see that there are two bilateral columns of cells that are really important for breathing, including housing the respiratory rhythm generator known as the Prebutzinger complex. So this complex is located bilaterally, and breathing rhythm begins within this complex. It's then transmitted to relevant respiratory related motor neuron pools, including the phrenic motor neurons located in the cervical spinal cord. And it's their axons that form the phrenic nerves that innervate the diaphragm. I introduce all of this circuitry not to bore you, but because in my talk today, I'm going to focus on activity directly measured from these phrenic nerves, as well as rhythm that begins within the medulla. I'm focused on two specific hypotheses today. The first is that neonatal systemic inflammation leads to lasting impairments in adult respiratory plasticity by changing adult microglia. The second is that maternal opioids leads to impairments in neonatal respiratory control and neonatal breathing. But first, I want to talk a little bit about the lasting effects of neonatal systemic inflammation on adult plasticity. A little bit of background about plasticity. Plasticity can occur anywhere within the central nervous system. One of the most well-studied forms of plasticity occurs within the hippocampus in the form of long-term potentiation and is believed to be really important for learning and memory. Another well-studied form of plasticity occurs in the spinal cord in the form of sensory hypersensitivity or chronic pain. I want to emphasize that this form of plasticity occurs in the dorsal or sensory component of the spinal cord, whereas the plasticity I'm going to focus on in my talk today occurs in the ventral or motor aspect of the spinal cord. For the purposes of my talk today, I'm defining plasticity as a persistent change in behavior or activity based on prior experience. The respiratory system was once thought to be pretty hardwired and immutable to help maintain that homeostasis in the body throughout life. But we now know that it needs to exhibit significant plasticity to be able to learn and adapt and appropriately control breathing. There are a few examples where we think respiratory plasticity is likely physiologically important, including during pregnancy, over the course of development and aging, ascending to altitude, and after disease or injury. So these are just a few examples of why the respiratory system needs to be plastic. It needs to learn and adapt to appropriately maintain breathing. One of the most frequently studied models of respiratory plasticity is long-term facilitation, and you'll see it abbreviated in my slides as LTF. So this is a form of respiratory plasticity that's induced by acute intermittent hypoxia or 3 episodes of low oxygen separated by periods of normoxia. Upon returning to normoxia, we can then monitor changes in neural activity or behaviors such as breathing over the subsequent 60 minutes. In a rodent model, we can directly record from the phrenic nerve, and after amplifying, filtering, rectifying, and integrating, we end up with these phrenic neurograms, which look like this, where each of these upward deflections is the neural correlate of a breath. We can then compress them over time and look at how neural activity is changing. Those compressed phrenic neurograms look like this, where you can see that there's an increase in phrenic neural activity in response to each of these low oxygen or hypoxic episodes, and that upon returning to normoxia, there's a progressive increase in phrenic neural activity compared to baseline, which is denoted by the dotted line. This change from baseline is what we call long-term facilitation and is a form of plasticity since the stimuli is no longer there, but there's been a change in that neural activity based on the prior experiences of these hypoxic episodes. This has been a really powerful model for us to better understand plasticity within the respiratory system because it allows us to isolate changes within the neural activity that are independent of the mechanics of breathing and really see how the nervous system might be changing. So acute intermittent hypoxia can evoke plasticity in a number of respiratory related regions associated with breathing, but this form of plasticity is not just occurring in respiratory control regions. We know that acute intermittent hypoxia can also induce plasticity in circuitry associated with grasping and locomotion, as well as ankle strength. This form of plasticity is also being tested therapeutically to increase ankle plantar flexion strength and increase walking speed and endurance after incomplete spinal cord injuries. So there's lots of potential for evoking this form of plasticity in therapeutic interventions to promote more motor activity, both within respiratory circuitries, but in other motor circuitries as well. But for us to really understand both the physiological role of the respiratory system being plastic, as well as being able to use it therapeutically, we also need to understand the ways in which this form of plasticity can be undermined. And we know that this form of plasticity can acutely be undermined by inflammation. Inflammation is associated with almost every disease and disorder you can think of, including the ones that are highlighted here. But as I alluded to at the beginning of my talk, we're specifically interested in the effects of early life inflammation. Early life inflammation is actually quite common. 65% of premature infants experience at least one infection during their hospital stay, which makes them 50% more prone to apneic events or events where the infant isn't breathing. And it causes a significant portion of deaths within the first week of life and accounts for approximately a third of all neonatal deaths. So not only is early life inflammation quite common, but it can have some pretty serious consequences. And we are investigating the acute effects of early life inflammation for breathing, but we're interested in what the lasting consequences are for early life inflammation. We know that in other non-respiratory regions, there are lasting consequences of early life inflammation, including by blunting adult immune function, impairing other forms of plasticity, such as hippocampal dependent learning. And it also increases the incidence of neuropsychiatric disorders and age-related cognitive decline. So there can be lasting consequences for this early life inflammation. And we're interested in understanding what the consequences are for respiratory control. Our fundamental hypothesis is that neonatal systemic inflammation leads to lasting impairments in adult respiratory plasticity in the form of long-term facilitation, and it does so by impairing adult microglia. So why might early life inflammation be having any of these lasting consequences? To start to understand that, we need to talk a little bit about some critical periods during development. So if we're looking at here is a timeline for development of the rodent, where E0 is day of conception and P0 is day of birth. If we just add in some of the important developmental events for the nervous system, we can see that there are a number of critical events that must occur in utero for the central nervous system to function at birth. But it remains immature and it continues to mature over the subsequent days and weeks. If we add in development of the respiratory system, we can similarly see that there's a number of important events that have to occur in utero, and the respiratory system has to be fully functional at birth for breathing to occur. but it again remains immature, and there's a number of important maturational events that happen over the subsequent days and weeks. So we're interested in understanding what happens when you stress the system by challenging it with inflammation early on in life when the maturation of these systems still remain immature. And does that lead to any permanent changes that last into adulthood? So why might activating the immune system be doing anything to the central nervous system? The central nervous system was once thought to be pretty immune privileged from what happens peripherally, but we now know that there's substantial bidirectional communication between the peripheral immune system and the central nervous system. Activating the peripheral immune system increases cytokines, which through a number of different mechanisms, including active transport across the blood-brain barrier, and activation of vagal afferents, will activate the central nervous system. There are two primary cell types that respond to these type of stimuli, and those are microglia and astrocytes. Microglia and astrocytes will then signal to neurons, leading to changes in neural activity and ultimately contributing to changes in behaviors. Microglia and astrocytes will also activate the hypothalamus, and collectively, the central nervous system will then feed back onto the peripheral immune system and help to lead to resolution of the stimuli that activated the peripheral immune system. So in our studies, we're evoking our understanding of this communication. We're activating the peripheral immune system with lipopolysaccharide, or LPS, a component of gram-negative bacteria. And then we're monitoring what are the lasting changes in respiratory control regions within the central nervous system into adulthood. So using this model of neonatal inflammation, we're going to activate the immune system in the neonate early on in life. And what we find is that there's an initial increase in mortality following neonatal inflammation. Importantly, there was 0% mortality in our control or saline injected neonates. And further, there's heightened male vulnerability early on in life to this early life challenge. And this idea of heightened male vulnerability early in life is actually quite common, though the mechanisms that contribute to and lead to that remain poorly understood. And we are interested in better understanding this early life male vulnerability. But if we follow those animals that survive into adulthood, we don't see any major changes in weight gain in males or females between treatment groups, suggesting that this early life stressor that we're challenging them with isn't completely derailing their overall development. But as I mentioned, we're interested in the effects of neonatal inflammation on adult respiratory plasticity in the form of long-term facilitation. So if we look at LTF in the adults, in our adult male controls, as one would expect, we have normal development of long-term facilitation. However, in our adult males, after neonatal inflammation, we have abolishment of adult respiratory plasticity. And you can see the group data shown here on the right. We've also looked in adult females. Our adult female controls have normal development of long-term facilitation. But we have, again, abolishment of adult respiratory plasticity in females after neonatal inflammation. So somewhat surprisingly, just one bout of sickness early in life leads to this lasting impairment in adult plasticity. We then wanted to understand what's causing this impairment in plasticity. And we hypothesized that maybe this early life inflammation is leading to some sort of ongoing inflammation into adulthood. To test this, we acutely manipulate adult inflammatory signaling by acutely treating with a non-steroidal anti-inflammatory drug, ketoprofen. In our adult male controls plus acute ketoprofen, as one would expect, we have normal development of long-term facilitation. In our adult males after neonatal inflammation plus acute ketoprofen, we now have restoration of adult plasticity. And again, the group data here on the right. We've also looked at this in adult females. Our adult female controls have normal development of plasticity. And similar to the males, we have restoration of adult plasticity after neonatal inflammation with acute ketoprofen. So these results strongly support that there is some sort of lasting inflammatory signal into adulthood that's contributing to this loss of adult plasticity. So you may now be asking yourself, okay, great. So what is this inflammatory signal? And I'll save you the suspense and tell you that we don't yet know. We do know that there is an increase in inflammatory gene expression in neonates acutely after the inflammatory stimulus, but that the adults return back to baseline levels. So there's no lasting increase in inflammatory gene expression into adulthood in respiratory control regions. But there's an important caveat to these results, and that's that we looked at homogeneous gene expression. Homogeneous gene expression includes all different cell types within the central nervous system, including the 3 that I've highlighted here. As we talked about earlier, microglia are the primary first responders to inflammatory stimuli, but they make up only around 5 to 10% of cells in the central nervous system. So if microglia are changing in response to early life inflammation, we'd be unlikely to see that change by just looking at homogeneous gene expression. So instead, we've moved to looking more at isolated adult microglia after neonatal inflammation. Microglia are known to be the resident immune cells of the central nervous system, and they respond to inflammation in many different ways, including by altering their morphology and increasing phagocytosis to increase the eating of debris within the central nervous system. They can also increase in number through proliferation and migration, and they can increase inflammatory related signaling. So we've been looking at changes in adult microglia after neonatal inflammation by assessing adult microglia number, as well as adult microglia transcriptomics. If we look at adult microglia number, we see that there is an increase in the number of microglia in the male medullars compared to control. While this trend was evident in females, there was no significant difference between groups, and there were no significant changes in the spinal cord in either treatment group or sex. So just one bout of early life inflammation leads to more microglia in the adult male medullars. If we look at microglia transcriptomics in both of our treatment groups, so adults after neonatal saline or neonatal control, and adults after neonatal inflammation, and we immunomagnetically isolate the microglia, there's an increase in inflammation and neurodevelopmental related genes, including genes that overlap between these 2 categories. If we break this down by sex, we can see that there are a number of genes in common between the 2 sexes, but some that differ between females and males, with females having more differentially expressed genes than males. So where does this leave us? Well, we know that neonatal inflammation is leading to abolishment of adult plasticity, and that we can restore plasticity by acutely manipulating inflammatory signaling. And we think that microglia are likely key to this abolishment of adult plasticity. There's more male medullary microglia and more differentially expressed genes in females. So it's suggesting that maybe males and females actually are initially responding to this inflammatory stimuli differently, contributing to maybe differences in the mechanisms that contribute to abolishment of plasticity, which I think really emphasizes the importance of making sure that we study males and females. Even though the phenotype might be similar, there might be differences in the mechanisms contributing to that phenotype. And to develop better treatments, we really need to make sure we're studying both sexes. One story that I'm not going to tell you about today is that we also have evidence for the involvement of astrocytes in impairments in adult plasticity. We have other evidence that shows that the neurons themselves are not impaired by early life inflammation, but instead their activity is affected by changes in microglia and astrocytes. So there's really important differences in how these cell types communicate with each other to lead to changes in these neural pathways that control breathing. So I've spent the last few minutes or so telling you about our story related to neonatal systemic inflammation. I'll pause now just to see if there's any questions and then move on to talk about maternal opioids. And I know it takes a minute to type, so I will give folks another second or so.
folks would also like to drop their response in the chat or Q&A, that would be okay too. And Adrianne, maybe could you repeat the question and I can also drop it in the chat?
Yeah, so I just wanted to see if anyone had any questions about the first half of the talk, and then I'll switch into the opioids, which I know most people are probably interested in. So it's also okay if there are no questions. There should be more time for questions at the end too.
We can also do a raise hand if you want. Yeah, are you comfortable if folks also like raise their hand throughout, or would you like to wait to the end?
I'm totally flexible. I just wanted to this is like a natural break, so I thought I would pause in case there were any burning questions related to neonatal inflammation.
Yeah.
It looks like maybe there's not. That's also okay. Okay, well, I'll charge forward just so everyone doesn't have to sit in that awkward Zoom silence for too long. And then if there are any other questions, there will definitely be time at the end. Okay, so let's transition a little bit more into talking about the effects of maternal opioids on neonatal respiratory control and neonatal breathing. As I'm sure many in this audience are aware, the opioid epidemic continues in the US and globally with an increasing number of opioid-related deaths. Yet one understudied population on the whole, though clearly not with this group, are the effects on pregnant women. There's been also an increase in the amount of opioids consumed during pregnancy, with more than a 4-fold increase in maternal opioid use reported at the time of delivery. Recently, the National Institute on Drug Abuse reported that 14 to 22% of pregnant women have received an opioid prescription, with this statistic being as high as 42% in some regions of the US. This is resulting in approximately a baby being born every 15 minutes having been exposed to opioids in utero. So there's clearly an increasing population of pregnant women using opioids, resulting in an increasing population of infants born after in utero opioid exposure. Pregnant women who receive an opioid prescription are often prescribed methadone, buprenorphine, or morphine. And infants born after in utero opioid exposure are often diagnosed with neonatal abstinence syndrome, or NAS, also known as neonatal opioid withdrawal syndrome. And these infants have diverse symptomology, including respiratory distress, tachypnea, yawning, sweating, GI distress, tremors, and a high-pitched cry. It's thought that these symptoms result from dysfunctions within the central and autonomic nervous systems, as well as the GI system, though the etiology of many of these remains poorly understood. Further, there's no consistent or accepted standard of care for these infants. A lot of improvements have been made on non-pharmacologic interventions for these infants, but many still require other exogenous treatment. There's also been few controlled studies on the outcomes or treatments for these infants. So we're interested in better understanding how do maternal opioids lead to changes in respiratory related networks and contribute to changes in breathing. If I revisit this diagram of critical periods, there has been work in animal models showing that opioids beginning at or before conception impair neurogenesis and also impair myelination broadly within the central nervous system. There was one study that showed that early life opioids also led to reorganization of respiratory networks and likely contributes to some breathing deficits. We're taking a unique approach to studying the effects of maternal opioids and beginning maternal opioids late in gestation. This is intentional. We're beginning opioids after important elements of the respiratory control network. Those cells have been born, they have migrated to their final location, and they have made connections to their appropriate targets. So what we're interested in studying are the effects of maternal opioids on maturation of this system, just as it becomes rhythmically active in utero, and as it makes this important transition from the in utero environment to air breathing. So we're interested more in the effects of opioids on maturation of this neural circuitry rather than genesis of the neural circuitry. If I also revisit this diagram I showed you earlier of some of the respiratory control networks, I gave you a slightly oversimplified version of this circuitry, and I want to revisit that here briefly. So as I mentioned previously, the Pre-Butzinger complex is an essential respiratory rhythmic generating center and is believed to be the putative control center for inspiration. However, early on in life, there's actually a second respiratory oscillator known as the parafacial respiratory group. And these two oscillators have substantial communication between each other, contributing and promoting neonatal breathing. Interestingly, early on in life, and actually throughout life, these two oscillators dramatically differ in their opioid sensitivity. The Pre-Butzinger complex is highly opioid sensitive, and opioid-related deaths are often associated with opioids silencing this respiratory rhythm generator, whereas the parafacial respiratory group is highly opioid insensitive. So what we're studying is how do late gestation opioids, after these networks have already developed, but as they are just beginning to rhythmically communicate with each other, how might maternal opioids influence development of this communication between these two important respiratory oscillators for breathing? Our experimental paradigm includes injections that begin at embryonic day 17, which is just as those respiratory networks are becoming rhythmically active, and then continuing through that transition from the in utero environment to air breathing in the subsequent postnatal days. We're using methadone as our opioid of choice, since methadone is frequently prescribed to pregnant women, and then we're monitoring neonatal breathing with whole body plethysmography. We have a control group receiving maternal saline injections, and again, we're going to monitor breathing in those neonates. We also have a second control group, which is a maternal no treatment group, to control for the potential stressful side effects of repetitive injections during gestation and the stressful effects that might have on neonatal breathing. What you're looking at here is a plethysmograph trace from a neonate after maternal no treatment. This neonate has relatively stable breathing with the odd apneic episode, so the odd episode where the infant is not breathing. And that's relatively common early on in life. We see something similar in a neonate after maternal saline, relatively stable breathing with the odd apneic episode. However, in neonates after maternal opioids, we see more instability in this baseline breathing and more frequent and longer duration apneic episodes. So they have less stable breathing and they have more periods where they're not breathing at all. We've looked at neonatal breathing from the day that they're born at P0 through to postnatal day 5. What we find is that there's an initial increase in the number and duration of apneas right after birth, but that they seem to catch up to their control peers by postnatal day 5. And so we're interested in better understanding what's changing in those neural networks that allow them to recover and catch up. And does this create infants that are acutely more vulnerable due to their instability in breathing early in life after maternal opioids? We've also looked at what happens when you challenge the respiratory system with normal respiratory stimuli. What I'm showing you here is the responses to low oxygen or hypoxia. In a neonate after maternal no treatment, it experiences this normal biphasic response. So there's an initial increase in breathing during phase 1 of the hypoxic ventilatory response, and followed by the hypoxic ventilatory decline during phase 2. And in neonates, this extends below normal baseline breathing. We see something similar in neonates after maternal saline, this characteristic biphasic response. However, in neonates after maternal opioids, we see a blunting of this phase 2 of the hypoxic ventilatory decline. We've looked at this from P0, so day of birth, through postnatal day 5. We don't see any significant differences between treatment groups for phase 1, but we see this blunting of the hypoxic ventilatory decline during phase 2 at postnatal day 0 and postnatal day 1. We don't yet know whether a change in the hypoxic ventilatory response is actually a good thing or a bad thing for the neonate, but we do know that the response exhibited by neonates after maternal opioids more closely resembles the adult hypoxic ventilatory response than the neonatal response. But whether that's a helpful adaptation or not, we're still investigating. So where does that leave us? I showed you some data demonstrating an increase in the number of apneas. We also know that the duration of apneas increases. We also see an increase in the number of post-SI apneas, though I didn't show you that data today. And we have a blunted hypoxic ventilatory decline. All of these together, we think, are indicating that these neonates have a destabilization in their breathing. And we think that this destabilization is mediated by the effects of opioids on how these two respiratory oscillators are communicating with each other and the maturation of this communication between these respiratory oscillators. But as many of you might know, there are many of the infants that are born with NAS also require additional treatment. These infants are encouraged to continue breastfeeding, and there's a debate about how much opioids is actually getting through the breast milk. But many of the infants also require additional pharmacologic interventions, which usually includes repetitive exogenous opioids. These infants are given similarly methadone, buprenorphine, or morphine. Though there's no standard dose or dosing regimen for how much opioids to give and when to give them. So we've been further studying what happens to breathing when you add additional opioids to the system after maternal opioids. We're using the same experimental paradigm with injections beginning at E17, which is just as those respiratory oscillators are becoming rhythmically active, and then continuing through this transition to air breathing. But now we're layering on neonatal opioids beginning at the day of birth and continuing through postnatal day 5. We're using methadone as our opioid of choice and then giving repetitive neonatal methadone after birth. We have and monitoring breathing with whole body plethysmography. We're also using a maternal saline group where again, those neonates are going to receive repetitive opioids after birth, and a maternal no treatment group whose neonates are also going to receive repetitive opioids. So what you're looking at here again are more plethysmograph traces from neonates after maternal no treatment or maternal saline. They have relatively stable breathing at baseline, like we talked about previously. We know that opioids inhibit rhythm generation by inhibiting that Prebutzinger complex I mentioned previously. So we would expect that acutely in response to additional opioids, there's an opioid-induced respiratory depression, and that's what you see here. It's a slowing in breathing in response to subsequent opioids. So this is the normal response to opioids. In neonates after maternal opioids, we have more instability in their breathing and more frequent apneic episodes. In response to subsequent acute opioids, we see a blunted opioid-induced respiratory depression. In other words, breathing is maintained despite being faced with additional opioids. If we look at how this changes over development, we can see that this blunted opioid-induced respiratory depression is really occurring at postnatal day 0, so the day of birth, and 1 day later, but they seem to catch up over the subsequent days. If we take out the control groups from this for just a second, we again see that there's this blunted opioid-induced respiratory depression early, but then this rapid sensitization to opioids that happens at postnatal day 2. And so we're interested in better understanding what causes this rapid sensitization to opioids at postnatal day 2, and what does this reflect about maturation of the respiratory circuitry. So where does that leave us? Well, we have evidence that maternal opioids leads to acute destabilization of neonatal breathing, and that it causes a blunting of the opioid-induced respiratory depression. And we have other evidence that supports that maternal opioids is actually delaying the maturation of the Prebutzinger complex and increasing reliance on the parafacial respiratory group. So that's a fundamental change in how these respiratory networks are functioning and maybe contributing to additional vulnerability for these neonates early in life. But we're also interested in what are the lasting consequences of this change in respiratory circuitry into adulthood. So if I start to wrap up, my talk today is really focused on the lasting effects of neonatal inflammation for respiratory control and breathing. And I talked a little bit about some of our early work looking at how maternal opioids can lead to some acute impairments in neonatal breathing. And we think it's doing that by fundamentally changing the circuitry that controls breathing. Ultimately, we think that these early life stressors are changing how distinct cell types within the central nervous system communicate with each other to lead to changes in neural activity and contribute to changes in behavior, such as breathing. I didn't talk much about microglia or astrocytes in our opioid model, but we do have some evidence that they are also being changed by early life opioids, and that may be contributing to some of these breathing deficits that we see acutely. And with that, I want to thank the awesome team that I have in the lab. I showed you some data today from Sarah Baylor that's contributing towards her PhD thesis. Sarah's project's really looking at how neonatal inflammation changes adult microglia. Robin Naidu is a graduate student in the lab, and she's really looking at how both viral neonatal inflammation might also lead to lasting impairments in breathing, and what happens when you challenge adults again with another inflammatory stimuli. Amanda Johnson's a new graduate student in the lab who's likely going to be working on the maternal opioid project. And Emily McDonald's an undergraduate in the lab who's looking at how opioid receptors change after maternal opioids. I also showed you some data from Austin Hawker's PhD thesis, as well as Nina Morrison's master's thesis. And I want to acknowledge our amazing team of collaborators. We have a longstanding collaboration with Dr. Jody Waters at the University of Wisconsin-Madison, looking at adult microglia. And we have ongoing collaborations with Steve Johnson at UW-Madison, Carrie McCurdy here at the University of Oregon, and Matt Hodges at the Medical College of Wisconsin. And of course, we couldn't do any of this without funding, including funding from the NIH, as well as a seed grant from the University of Oregon. And if anyone is looking to foster some new collaborations or has some new ideas or is interested in joining our group, we are always looking for new people to connect with and work with. Feel free to visit our lab website or email me directly. And with that, I would be more than happy to take any questions.
Thank you so much. I actually had a question for you. Have you considered potentially looking at sort of normal use patterns that might occur during pregnancy and how those influence the trajectory of respiratory functioning, like, you know, heroin use early on, and then how changing to methadone, you know, at some point in the pregnancy changes things, or yeah, and also drug interactions with things like tobacco.
Yeah, I love that question, and I would love to do that. We are just kind of getting this project off the ground, but I totally agree with you that the late gestation model probably isn't mimicking normal use from pregnant women, though there is some evidence that pregnant women begin opioids late in life. But I totally agree with you, like looking at the combination of different opioids at different developmental stages, I think would be really interesting and may be having even more profound effects on that developing respiratory circuitry. And I think one of the really powerful tools in an animal model is that we can start to layer on some additional polysubstance use and things like that in a really controlled way to start to tease out what are opioids themselves doing, and then what does the combination, so are there additive effects of the different pieces? I think that is like one of the real benefits of the animal model, because we have so much control over some of those factors. But yeah, I would love to do all of the things that you mentioned, and I think that would be really helpful and informative. If I had to hazard a guess, I would guess that we're probably going to see more profound effects as we start adding in more opioids and for longer duration. And so this was more of a like, let's just see what happens to the circuitry piece and then start adding in more of these factors to get a better idea about what might be contributing to some of these human infants that are exposed to opioids for longer periods. Yeah.
Yeah, that's exciting work. And that's exactly what I was thinking about animal models as you were presenting. I was like, oh, wow, there's just so many great opportunities to really isolate the effects one by one that we certainly don't have with humans.
Yeah, definitely.
Yeah,
so I would love to talk to all of you more about like, what would you, if you had to pick one other thing to add, like what would be the thing that you would add? So yeah, I'm open to all of those suggestions. So please feel free to share.
That's a great question for the group. I was also going to ask you about like social environment that the rats are living in. There's that, you know, I think famously in the substance use literature, even though it was a very small study with some limitations, the rat park study that people that might be familiar with, where they looked at the environmental influences of cocaine uptake by rats. And so, yeah, I was just curious how you control the environment, and what does that usually look like in these situations? Yeah.
Excellent question. It's really interesting. We were initially really worried about sort of maternal neglect with treating after the neonates are born. And there's definitely, I would suspect that given the behavior of the rats when they're getting opioids, I would suspect that there is some of that, which is part of the reason why we had the 2 additional control groups. So if it's just the injection, we should see that with our saline group, or if it is we don't really see any differences between our maternal no treatment and maternal saline groups. But there is literature that supports that maternal separation models can also induce some pretty profound changes in not only respiratory circuitry, but many different regions of the brain. So we don't separate the neonates from the mums at all. And we are trying to think of ways that we can better quantify whether maternal behavior is different. And so we've thought of some cross-fostering models where the neonates were exposed to opioids in utero and then ended up with a mom that got maternal saline or maternal no treatment, to start to look at whether that might be influencing some of the pieces that we're seeing. We haven't yet done those. We've definitely had some inquiries from reviewers and things like that about whether that's in our plan or not. I would like to do it. I think it would be really important to understand sort of how that maternal behavior might be influencing some of these pieces. So yeah, excellent idea. I wish I had a better answer for you.
No, that's super interesting. It also makes me think about, and then I will let other people ask questions. And for those of you who don't know, I think for the webinar function, you can, in the Q&A, you can drop questions in or raise your hand, and we can allow you to speak as well. So feel free to do that. But it makes me think about, child welfare removal. And so like it actually could be a potentially useful comparison to look at all of the rats as potentially opioid affected, and then moderating their environment or modifying their environment. I don't know, do rats have similar behaviors as other animals, where you can maybe induce some sort of maternal behavior in for rats that didn't give birth to that specific pup?
Yeah, so we can definitely cross foster really quite easily. They are really, depending on depending on the rat, it definitely can be rat specific, sort of, I would imagine, like any animal. But they are really tolerant to taking on additional animals that were not theirs. So we can definitely do a lot of those studies where we can influence the environment. We can also influence the amount of enrichment they have access to, the amount of light, and other aspects of the environment as well that may be contributing to all of these different pieces. So yeah, we can definitely control for a lot of these, and the rat model is pretty tolerant to allowing us to mix the neonates from different treatment groups, which I think could be really powerful.
Cool. Very cool. Thanks for sharing. Yeah. Are there other questions that folks have?
Maybe I just put everyone to sleep thinking about breathing.
I think that it is so useful to have so much information about these specific systems, and I think that students or those who are joining maybe from the psychology department might have some exposure to this, but certainly students from other places on campus might have less exposure. And so yeah, I think it's just so useful and so much information too. So, but I'm not seeing anybody have additional questions, but can folks reach out? I guess if people have additional questions, maybe they can reach out to Ashley and she can collect them and send them your way.